Hello, students. I'm seeing many of you have joined already. We'll wait for a few minutes for the other people to join and resolve their issues if there are any. And then we'll start the class. Can you guys hear me? You can send a message.
Yes, ma'am.
Good, thank you. I'm just waiting for 3-4 minutes so that other people can also join. Then we'll formally start. Okay, so Okay, so I think many of the students have joined till now. I am in a minute. I am trying to share with you link of the book, guidance textbook of medical physiology. I got a copy of that, soft copy. So I want to share that with you. In a minute, I'm trying to share it so that you can get your study material from which you can study your all of the things that we are going to study in this course. So let me try it. As the file is larger, this will be a Google Drive link. I'm trying to give you the rights for that, but if you need permission, you can send me an email for permission. Then I'll allow it. Okay. So here I'm sharing the Drive link for guidance textbook of medical physiology. This is your textbook for most of the part of the course. You can access this through Google Drive. Okay. Now we should start with the next part of the lecture. So I think now most of the people have already joined. So we let's briefly review the previous class, what we have done in the previous class. So we have covered two parts basically, propagation of action potential and the properties of action potential in the previous class. Let's slightly review it, and I think it will be difficult to ask questions to you here. So I prefer not to ask questions, but yeah, I prefer not to ask you questions, but simply review it myself. So let me share my presentation. Okay, so can you guys see me my presentation? Can you guys see my presentation now?
Yes, ma'am.
Yes,
Okay, great. So this is the thing that we have done last time. Let's review it. We talked about action potential first, and then propagation of action potential. So action potential is basically the change in the potential difference across the membrane of a neuron or any excitable cell that causes the cell to be excited. So the cell comes in an excited state, and that time it can send a message. Now, the second phenomenon that is associated with that is propagation of the action potential. So, propagation mean we can say in more simpler terms movement or spread. So, an action potential listed at any point on an excitable membrane usually excite the adjacent portions of the membrane, resulting in propagation or what we call spread of the action potential. And what is the mechanism of the spread? We talked about it in the last class. That basically the positive electrical charges are carried by the inward diffusing sodium ions through the depolarized membrane, and then they move actually several in both directions along the core of the axon till they reaches axon hillock. Now they actually increases the voltage of for a distance of about 1 to 3 and so on. So sodium channel in these areas immediately open, and an explosive action potential it arrives. This transmission of depolarization process along a nerve or sometimes a muscle fiber. This is sometimes also called nerve or muscle impulse. If it is a nerve, this will be nerve impulse. If it is in muscle, This will be called a muscle impulse. This figure we talked about it. Briefly revise that. If this is a neuron, example of a neuron, on which one stimulus arrives at this point, what will happen? That sodium channels open, so sodium ion influx is there, and those ions then move in both directions. If it is cell body or if it is dendrite, in both cases. If this is axon, this is generally unidirectional. So here it is how it's spreading more and more, and ultimately, if this thing continues, then the whole membrane will be depolarized. That is, change from negativity to positivity. And direction of propagation. An excitable membrane has no single direction of propagation except for axon. But the action potential travels in all direction away from the stimulus. See, normally we said that mostly if we talk about some physics of this, then mostly this is due to repulsion of positive charges towards each other. So when all the sodium ions are coming in, they close to each other, and positive charges they repel each other so that they move away from the stimulus site, and even along all branches of nerve fiber and so on. Then we talked about properties of action potential. We talked about these 5 properties: all or nothing principle, also called all or none. That when it reaches the axon hillock, the message will be sent. If it does not reach the axon hillock, then no message will be sent. So this is I or O, on or off. Just for a minute, let me see if someone want to admit inside. Is there anyone in the lobby? I think I cannot see it. Students can see it. If someone is there and trying to admit into the class, please tell me, okay? If you know it. Uh, just for a minute. No, I think all are in. Okay. I think all are in. All right. Then, second thing that we talked about is summation. Which means that when more than one stimuli are arriving, then both will naturally move towards each other, because we know, see, just we talked about that both are moving in all directions. So when they are moving in all directions, they will somewhere come close to each other. It means that sodium ions come close to each other, and of course both will merge. So mostly their effect will be merged. Now most of the times this is exciting membrane further. Sometimes it may be different case. We may about it later when we talk about memory and learning. So latency. This is simply the time period in milliseconds from initiation to the peak. When stimulus arrives, channels open to the peak reaches. Peak means 0 or plus 35 reaches. This small time period is called latency. It varies from cell to cell. Then refractory period, we said there is a break between two action potentials. That is the period in which no action potential could be generated on a membrane, even if a stimulus arrives. Now, this refractory period is precisely due to, we said that for 2 reasons. As first reason is this, that all sodium is already inside because of action potential, and second, that ion channels, the sodium channels are inactivated, due to which no action potential can be generated. So last thing that we talked about in last class is accommodation. The modification or slowing down or speeding up of a response to a benign stimulus. When a stimulus arrives, and this is the cell is sending a message to the next cell, but nothing is happening, there is no response, then cells modify their behavior. That is, slow down the response. Maybe if there is more stronger reaction, then speeding up of the response, or maybe not even not giving any response. This is called accommodation, and this phenomenon is actually responsible for almost all kinds of adaptations regarding learning and memory. So almost all of the learning and memory, which we have heard of it most probably, that it is based upon reward and punishment. So based upon reward, it can speed up some response and maybe slow down some response. Based upon punishment, again, it can slow down a response or speed up a response. This is possible, and that is called accommodation. That is based for mostly learning and memory. So here we have finished. Now we go to the next section. That is myelination and nerve conduction. Now we talked about nerve conduction. That is a spread of a again action potential from one point to another inside the cell, and then moving through all of the axon, reaching the end bulbs, and then sending a message. uh, Now, few nerve fibers are small. For example, in CNS, brain is small, and within the brain, the neurons are also very, very small things, and mm, action potential have to move through only small distances, even microns probably. So even less than that. So what happened that those cells can message some transmit the message easily. But what happened that when the neurons are larger, like in case of large sensory neuron, which is, for example, moving from our toe till the spinal cord, it may be according to the height of the person, it may be 3 feet long. तो अगर 6 feet का person है तो 3 feet long लंबा हो सकता है fiber. And same goes for motor neuron, which is coming out of the spinal cord and going till toe of our nail of the foot, for example. It may be very long too. So for those neurons, this is a problem that nerve conduction may become very slow, takes time due to spread of the ions. Now, what happened? That adaptations found a solution to that. Is someone asking something? Is someone asking something? No? Okay. All right. So well. So to solve this thing, that traveling the impulse for longer distances, cells adapted uh, the myelination. Now, what is myelination? Uh, in a nerve fiber, mostly the large fibers. Precisely when we are talking about fiber, we are mostly talking about the axon. So, so the large fibers are myelinated. They are having a, you can say, a covering, a sheath around them. And the small ones are mostly unmyelinated. They are not having the covering or sheath around them. They are axons basically. They're axons basically. The average nerve trunk contains about twice as many unmyelinated fibers and myelinated. So if there's a nerve trunk, a bigger nerve, commonly we call it nerve, it is actually a nerve trunk that contains many fibers, like some myelinated and some are unmyelinated. And maybe sometimes all myelinated, maybe some are all unmyelinated. So this is possible. Now the central core of a typical myelinated fiber is the axon. Now what happened that inside the center is always the axon, and the membrane of the axon is the membrane that actually conducts the action potential. Now axon is filled with in its center with the exoplasm, which is, we can say, cytoplasm of the neuron, and this is a basically intracellular fluid. Simply, we can also call it cytoplasm. Surrounding the axon is a myelin sheath, and that is often much thicker than the axon itself. What happened that myelin sheath is much thicker sometimes than the axon itself, because it actually has to do to basically make a covering which have to increase the speed of conduction about once every 1 to 3 mm. Along the length of the myelin sheath is a node of Ranvier. Say about 1 to 3 millimeters. There is a node we call node of Ranvier. Now let's see in a diagram from right and whole. Um, Here you can see this is the axon. See in between in yellow color, you can see that the sheath which is surrounding it is called myelin sheath is actually thicker than the axon itself. So it is basically insulating it. We can also say that this is basically insulating it. It's wrapped around it. Here you can see the layers which are formed, and then a fluidy material between among the membranes and the layers. And then there is a nucleus. It means that this is cell. So the axon is surrounded basically by one or sometimes more than one cells, which are wrapping around it in these membranes. And you can see there are layers, and there is fluid inside. This cell, which is making this myelin sheath, is called Schwann cell. On the name of the scientist who discovered it, the Schwann cell is having, here you can see a nucleus, and then all these layers which are filled with the fluid. This is the Schwann cell cytoplasm. Here you can see this, all of it. So it is making an insulation around the axon. Now, what happened due to this insulation? That this area, you can see ion channels are covered because this cell is wrapping around very closely, except for this small part which is uncovered. What happened that here are only sodium channels. Here there are, if sodium channels are there, normally not there, they're there, they cannot act. So when sodium enters at these points, the nodes of Ranvier, then what happened? That this sodium ion, all of these sodium ions which are arriving inside, actually moves along or jump along this whole part, which is almost free of the ion channels which are active, move from this whole place very fast and reach the next node. From here, we can say the sodium ions comes inside and then jump from this node to the next node. And on the next node, more sodium channels open, more sodium enters in, and then it jumps again. So this actually increases the velocity or speed of the nerve conduction, just like current in running in the wire inside the electricity. Just like that, they're moving like. So this actually increases the speed of conduction, make it very fast. And this actually reduces the time also in which it reaches the particular distance. For example, if we touch something very hot, we quickly remove take the finger back. This is the speed which is required. If this is slow, if this myelin sheath is not there, then our response from our finger, which is recording that information, that this is something hot we have touched, has to reach our spinal cord, and spinal cord will send a message to motor neuron that retract the muscles and all the other parts so that we take the hand back. If this sheath is not there, this process will be too slow, and the result will be that in the meantime we retract our finger, take our finger back. In that time, we will hurt our finger a lot. But see, it doesn't happen normally. We quickly take our finger back. So this speed is required at many places, and that is all those neurons which are involved in that process, the sensory or motor, are myelinated in this way. Here you can see a nerve trunk. The nerve trunk, many nerves are there. And here you can see very large nucleus of a Schwann cell. The Schwann cell is a very big cell here. You can see, though all cells are not like that. Sometimes one Schwann cell is covering only one neuron. This is also possible. But here you can see, this is a nerve trunk in which in the center there is a Schwann cell, its nucleus, its cytoplasm, and you can see projections of its cell membrane. There are projections of its cell membrane, many of them. And then what happened that they are covering many neurons here, and surrounding. Now, some of these will be completely covered. If they're completely covered, they'll be completely myelinated. Here, few are unmyelinated, few will be partially myelinated, and so on. Some are partially wrapped here, some will be fully wrapped, some are exposed a lot. So this is kind of nerve trunk in which it's happening. What it is showing, the function of Schwann cell is to insulate the fibers. B, partial wrapping here, which we are seeing, partial wrapping of the membrane and cytoplasm of Schwann cell around multiple unmyelinated nerve fibers. Many are covered by single cell. So myelin sheath is deposited around the axon by Schwann cell. How? The membrane of Schwann cell. Now, this is something which is happening in an embryo. When the embryo is developing, at that time, neuron cell is just like another cell, is a rounded cell. But later on, it takes its shape. The cell body and dendrites are formed, an axon is formed. At that time, myelin sheath is also formed. This is deposited. Schwann cell, first of all, its membrane envelops the axon. It wraps around by, we can say, extending its cell membrane around the axon, take the turns. This cell then rotates around the axon many times, laying down multiple layers of Schwann cell membrane, as you can see here. First, these layers are formed. Then Schwann cells develop a lot of lipid substance called sphingomyelin. This is basically insulating substance. So this substance helps in this insulation, indeed. This compound, sphingomyelin, is an excellent electrical insulator. It decreases the ion flow through membrane about 5,000 folds. 5,000 folds. This much it decreases the ion flow. So that's why ions move very fast from one node to another node. They jump. That is why we call this kind of conduction the jumping conduction also. Then the node of Ranvier. At the juncture between each two successive Schwann cells, if more than one Schwann cells are wrapping around it, a small uninsulated area about 2 to 3 micrometers is there only, where ions can still flow, and the movement is possible. I mean, movement of the ions across the membrane is possible. This is called node of Ranvier. So here only, as I have explained before, that the ion channels, they can open and messages will be transmitted. Is okay. Sorry, someone want to admit. Okay. All right. So we continue. Is there anyone else who want to admit? Let me check my email just for a second. All right. Okay. Sorry, I have to disturb for some time because what happened that some people are trying to admit coming inside the room, and I don't know. I have allowed everyone to come. Why it is happening? Someone is saying something. Mobeen.
मैम मैं बस यही कह रहा था कि एक अली रज़ाक जो है वो ऐड नहीं हो रहा ग्रुप में, वो मेरे साथ ही बैठा हुआ इसलिए।
हाँ, मैंने ऐड अभी किया है किसी को. आई थिंक दैट इज अली. इज ही एडेड नाउ?
थैंक यू।
Great. Okay. Okay. If there is a problem like that someone is want to enter and not able to enter, please send me a chat message. I'll get the message, will pop up on my screen and I'll see that. Okay. Thank you anyway. So we continue with that. We come back. Okay. So node of Ranvier we were talking about. So at the juncture between two successive Schwann cells, or if there is a single Schwann cell, there will be notch over there in a particular place, which will allow some current flow. So what happened that at the nodes, basically, the ionic flow is possible. Ions can come in or go out. Let's see in another diagram, which is showing saltatory conduction along a myelinated neuron. Now here you can see some more closer picture. So the axoplasm, this yellow lined area inside is the axon. This purple area is basically the myelin sheath, the myelin cell. And here small nodes are there. So this means this is one cell, this is another cell. Cell means the myelin, the cell which is making this myelin sheath, Schwann cell. This is another. And in between you can see the nodes. Now, then the ionic flow, you can see this unidirection from here to in this direction. Ions are moving from this direction to this direction. Now what happened that when on these nodes, ion channels, they open, like sodium channels, they open, sodium ions come in. When they come in once, like from here, they move very fast from this area to this area, because this area is insulated by the myelin sheath, the sphingomyelin, which is the compound. Then here, when they reaches here, more sodium channels they open, more sodium comes in, and then this jumps to the next place. Then next node arrives, and when next node arrives, then again more sodium channels they open, more sodium rushes in, and it moves again very fast. So this thing actually increases the velocity or speed of the conduction to several folds, several thousand folds indeed. This is called saltatory conduction, or this is also called jumping conduction. So saltatory conduction, and also it is also called, I'm writing so that we can record it, jumping conduction. This is very, very important in saving ourselves from any kind of hurt or a damage in any way, any case. So, well, this is something I have explained. Now, why this saltatory conduct? So let's define it. See, almost no ions can flow through the thick myelin sheaths of myelinated nerves. They can flow with ease through the nodes of Ranvier only. Therefore, action potentials occur only at nodes. We can also say that in axon, which is myelinated, an axon which is myelinated, action potentials occur only at the nodes and nowhere else. Yet, the action potentials are conducted from node to node is called saltatory conduction or jumping conduction. So, how we define it? Saltatory conduction or jumping conduction? This is action potentials conducted from node to node. This is called saltatory conduction or jumping conduction. So, the nerve impulse actually jumps down the fiber. You see, jumps down the fiber from cell body towards end terminals. So, from cell body towards end terminals. Which is basically the origin of term saltatory. Why called saltatory? Because it is jumping down the fiber. Now, what's the value of it? Why this is important? Well, there are two reasons. It is of value, but for two reasons. By causing the depolarization process to jump long intervals along the axis of the nerve fiber, this mechanism, of course, increases the velocity of nerve transmission from 5 to 50-fold normally in myelinated nerves, maybe more if it is a more thicker one. Now, saltatory conduction conserve energy. This is second very important importance of it. Number one is speed. Number two is energy. Because only the nodes they depolarize, allowing perhaps 100 times less loss of ions that would otherwise be necessary, and therefore requiring little metabolism for re-establishing the sodium-potassium concentration differences across the membrane after a series of impulses. Let's simplify it. We talked about that when this action potential passes, after that what happened, that sodium is inside, potassium is outside. Now that has to be re-established. I mean, sodium should go back out and potassium should come back in. Now, to resolve this, sodium potassium pump have to work a lot, because that is throwing 3 sodium out from inside and taking 2 potassium back from outside. Now, we know that for such one transmission, a sodium potassium pump need 1 ATP molecule. So one movement of ions, 3 sodium out and 2 potassium in, needs 1 ATP molecule. So it's very costly process in terms of energy. Now this saltatory conduction, as those ion channels are not opening, that much ionic transmission is not required, and same thing will happen, or same message will be transmitted through only fewer sodium ions. The re-establishing the resting membrane potential is required much less. It saves a lot of ATP to the molecule, to the cell, the neuron, the axon in precisely. So what happened, that this term is in terms of energy much less costly. That's why this is also adapted, and particularly when long nerve fibers will utilize lots and lots of energy, and it is saved in this way. So for two reasons, this process is very important, very useful. Number one, the speed of conduction. For example, if this is slowed, if it is not there, then what will happen? We may hurt our finger. See, जितने में हम टच करके उसको हटाएं, आहिस्ता आहिस्ता मैसेज जाए, उतने में तो हमारी उंगली जल जाएगी या हर्ट हो जाएगी बुरी तरह से. लेकिन क्या होता है, बहुत तेजी से मैसेज चला जाता है इसकी वजह से. So we are saved from damage. We can react or respond very fast. And secondly, saves a lot of energy. इतने सारे sodium channel open हो पूरे के पूरे axon, see, right from the spinal cord till the toe, what will happen? And it's two-way, like from sensory neurons, it's sending the information and then taking the message back through motor neuron. Then what will happen? That lots and lots of energy will be wasted, and that is all conserved due to saltatory conduction. So these are two evolutionary advantages due to which cells adapted this saltatory conduction or jumping conduction. And this was our main topic for today. One small thing, just some information to add, that velocity of conduction in nerve fibers, it varies too much. It varies a lot. It varies from as little as 0.25 meters per second in very small unmyelinated fibers to as great as 100 meters per second, the length of a football field in 1 second. Very large myelinated fibers. These are small unmyelinated fibers like in CNS. It may be 0.25 meters per second, but it works. What I want to add, that it works very well for them. Why? Because they have to travel a very small distance. Because see, how big is the brain? Big in terms of function, but not in structure. It's a very small thing, and neurons have to send a message to maybe micrometers away. So it works for them very well. But this is not enough for the functioning of large fibers like the one which is from our spinal cord to our hand, or spinal cord to the toe, or spinal cord to the any other part of the body. So in those, this small speed cannot work. So when they are myelinated, this velocity increases at approximately like 100 meters per second even. They can travel 100 meters per second. Anyway, so this is the evolutionary advantage, of course, a huge one. And that's why myelination is there and is adapted by the cell, is continued by the junction. So we end our today's lecture here. And if you have any questions regarding right from the action potential till myelination, I'm here to answer. I'm stopping my share, my screen, and coming back to you. Okay, so guys, oh, I also have to mark your attendance. In the meantime, you can make your questions if you want to. Okay, let me record your attendance first. Just 1 or 2 minutes for me. I'm recording your attendance from the list of attendees that is there with me. Okay, so I'm done with the attendance. Now, if you guys have any question, do you have? Is it clear? Is the topic clear to everyone? If you do not have a mic, you can send a message in chat.
Ma'am, can you show the slide with the axon?
Slide with what?
With the axon, myelinated axon.
Okay, sure. Yes, I can. Let me. There we are. Okay. Uh, is it the one you want to see?
No,
Can you see that now? Presentation?
I think it was on 17 or 18.
Okay, okay, okay. This one?
Yes. Could
17?
you explain this again?
Sure. Yes, why not? Um, what happened that this slide is showing the axon in the center, this yellow one, which is the lined area. And here in dark purple, it is showing the nodes of Ranvier. This, and in between here, you can see this is a two-dimensional diagram, of course, not a 3D. This one in the center is the myelinated cell, basically Schwann cell, which is wrapping around it. So this is one Schwann cell surrounding this whole area. The second is surrounding this whole area. Third is surrounding this whole area, leaving these areas where ion channels are can open upon requirement. So what's happening here basically that when, for example, on this point, stimulus arrives, sodium channels they entered through axon hillock to this area, right? Sodium channels open, sodium rushes in. When it rushes in, because this whole area is insulated, these ions moves very fast and quickly reaches the next node. When they reaches these nodes here, this node here, then sodium channels which are present here, all of them open. When they open, more sodium comes in. And again, next they are insulated. So they very quickly move in this direction. Again,
So they keep
same
on
process.
opening the other channels.
Yes. These on these nodes, they keep opening more channels, more sodium enters, and then there is a jump to the next node.
Okay.
So we
And
yes.
what about the signal? Does it travel on the Swan cells?
It travels these ions are actually what we call signal. Signal is basically what I mean, this is movement of these ions, just like flow of electricity in a wire. It's same.
Mm, okay.
So these are
And
all
what are
sodium
the
ions.
1, 2, 3 arrows?
Sorry, what?
The arrows with the 1, 2, 3.
These arrows are showing basically the insulation outside. The inside ones are showing ions. These arrows outside are actually showing the insulation, that this area is insulated. Then this small is left where ions can move. Then this area is again insulated. So it is showing 1, 2, 3 insulated areas.
Okay. Thank you, ma'am.
Yes, okay, welcome. Gee, anyone else have a question? जी और कोई सोचना चाहे तो सोच लें.
Ma'am, will these slides be uploaded on Moodle?
Yes, absolutely, absolutely. बिल्कुल ये Moodle पे I'll upload this lecture as well. This is recorded, but I'll also upload the PPT separately. PowerPoint I'll share separately. So there'll be PowerPoint and there'll be, then let me see if I don't know if I can have some, can show you some video of solitary conduction. I'm not sure, but if I find a good video, just for a minute, I have just to see one or two.
I have to stop and tell you guys.
In this video, I'll show you how neurons use a structure called myelin to speed up the transmission of electrical signals. Then, I'll give you one good reason why you should care a lot about myelin by introducing you to a different part of the axon. So, an action potential needs to be started here.
Oh, I think I'm missing in my record, but I'll what will happen that I'll just share a good video with you on Moodle. I have a few videos, and of course, we have to sort out which one is actually the correct one, because many people are explaining on YouTube or so, but some are explaining right, some are explaining wrong. So I have to find out from my records, and I'll share, inshallah, surely, video with you in some time on Moodle, a link or so. Iman is saying, can you explain formation of myelin sheath in the embryonic development? Yes, sure. So Iman is asking that formation of myelin sheath in embryonic development, how it happened. What happens that initially the neurons are just like all other rounded cell. Normally an animal cell is like a rounded cell. And what happened that later on they take their shapes. For example, if it is a muscle cell, this will elongate, then the sarcomeres are formed. If it is a neuron, just like that, if it is a neuron, then what happens? That this elongates in multiple directions, and some of the elongation, some of the, we can say, extensions of the cell membrane makes what we call dendrites, and one of the extensions make what we call an axon. Axon is longer. It is a longer extension of the cell membrane, and hence the cytoplasm goes inside it, and it makes a, we can say, tube kind of structure. And then after that, there are supporting cells in the surroundings, which are called these Schwann cells. And those Schwann cells start wrapping around after formation of that tube-like axon at that time. And then they wrap in multiple layers. When they are wrapped, that wrapping is complete. बिल्कुल ऐसे ही जैसे हम एक क्या कहना चाहिए कि कोई भी छोटी सी ट्यूब लें और उसके इर्द गिर्द हम कोई प्लास्टिक शीट को रैप करते जाएं, इसी तरह उसकी बहुत सारी लेयर्स बना दें, बिल्कुल इसी तरह से वो सेल अपने आप को रैप करता जाता है और लेयर्स बना देता है. When those layers are formed, then this cell, the Schwann cell, produces what we call the compound sphingomyelin. Sphingomyelin is a lipid substance that is having, of course, lipids have some insulating properties. So it is having insulating properties. Then this sphingomyelin fill in the membranes, and then it becomes like swollen with the help of that, which makes this structure that we see we have seen in this. Let me share that picture again. That's the one. Let's go back. The Schwann cell here. Then when this you can see between these membranes, the fluid is filled. So first of all, this part is present, only this yellow part. Then slowly this one layer is formed. Then this next layer is formed. Then this next layer is formed, and just like that, all the layers are formed. Then the cell produces sphingomyelin, and it fills in all the membranes and the layers to make this whole structure, and this insulates. And later on, it depends how much insulation is required. According to that, Schwann cells, they keep insulating the whole of the membrane, and ultimately when it is complete, then it becomes functional. This is how it happened in an embryo, basically. Almost all cells which are neurons, they are formed like this. Is it enough? Clear? Okay. All right. Anyone else do you have a question? No? Okay, so your attendance is already marked. So thank you very much. Inshallah, and I'll share this recording as well, share my PPT as well. I'll try to share a video of myelination or something with you too. Good one. And see you inshallah in the next class on Wednesday. We will continue with next part of it. That will be basically the sensory organs. We'll talk about sensory organs. Okay. Thank you very much. Class is dismissed. I'm ending the call.
Ma'am?
Yes. Yes, someone is asking something. You can leave the meeting if you are not having a question, okay? Ji. Someone is asking something.
मैम मैंने ये पूछना था कि ये गाइटन की बुक में से कौन सा चैप्टर है जो अभी हम पढ़ रहे हैं?
ये मैं अभी शेयर कर देती हूँ। ये 2-3 असल में 1-2 चैप्टर्स कंबाइंड हैं। उनमें से कुछ कंपोनेंट होगा और ये वाला जो है दैट इज़ प्रोबेबली नहीं, मैं शेयर कर दूँगी, अभी ऐसा ना हो मिस्टेक हो जाए। मैं अभी जब आपको पावर पॉइंट भेजती हूँ, तो उसके साथ ही मैं चैप्टर लिख देती हूँ उसमें, ठीक है? आई एम सेंडिंग यू पावर पॉइंट इन सम टाइम, मैं उसके ऊपर यूँ करूँगी, चैप्टर लिख दूँगी। पावर पॉइंट के ऊपर ही चैप्टर रीडिंग, पेज नंबर्स, ठीक है? फिर ईज़ी हो जाएगा सबको। ओके? अच्छा, जब बुक का लिंक खोलने में किसी को प्रॉब्लम हुआ तो फिर आप मुझे बता दीजिएगा। Any other thing someone want to ask?
मैम मेरे ईमेल को लिंक ओपन नहीं हो रहा था क्योंकि उसमें आ रहा था कि एक्सेस नहीं दिया हुआ आपको।
हाँ,
तो
वो
वो आप
आप
मुझे
मुझे
एक्सेस देंगे तो उसके बाद
हाँ, बिल्कुल, आप मुझे ना वो आपको पूछेगा कि एक्सेस देने के लिए एक ईमेल कर दें, तो आप मुझे वो उसको क्लिक करेंगी ना एक्सेस के लिए, तो मेरे पास आ जाएगा, मैं अलाउ कर दूँगी, देन यू विल बी एबल टू
जी मैम वो रिक्वेस्ट सेंड की ऑप्शन थी, वो मैंने आपको रिक्वेस्ट भेज दी, अब आप
कर
प्लीज
दिया?
उसको एक्सेप्ट कर लीजिएगा।
बस
जी
मैं
मैम।
अभी कर देती हूँ सारों की, सब लोग जो भी भेजते हैं ना, अभी मैं थोड़ी देर में सबकी कर देती हूँ।
जी थैंक यू मैम।
ओके, वेलकम। Okay, ji, so class is dismissed. Thank you very much.